So good morning. I am Nanfack Minkeu Ferdinand. I am working at the College of Worcester. And today my talk will focus on evolutionary function of seminal fluid adipokinetic hormone in Aedes aegypti. Aedes aegypti is a mosquito species, main vector of arboviruses like yellow fever, Zika, and other. So what is the seminal fluid? Seminal fluid are produced by the reproductive tissues of males and transferred to females during mating. And they influence the physiology of females, like oviposition, the feeding behavior, the fertility, and other. So they are mainly identified by the liquid chromatography and mass spectrometry. So as you can see in this image, at least 177 seminal fluid are found in Aedes aegypti. We know that there is a high evolution of seminal fluid, but unfortunately, their evolution is under study, or the impact of their evolution is under study. For that reason, we put in place our project that focus on a neuropeptide that we call adipokinetic hormone that was discovered for the first time in the seminal fluid of Aedes mosquito in our lab in 2014. That's insect neuropeptide that we call adipokinetic hormone has never been reported even today in the seminal fluid of any species. So the question is, what is doing in the seminal fluid? What is the function of that adipokinetic in the seminal fluid of any species? It is very interesting because this adipokinetic hormone, it is synthesized in the corpora cardiaca in the heart, and then it is mainly involved in the metabolism of insects. So if you check the literature, you see that one of the well-known functions of adipokinetic hormone is a metabolism function, mobilization of energy. So we know also that it's found in several insects. You can see, for example, it presents Inculex bpien, the main vector of West Nile virus. It is present in Anopheles gambiae, the main vector of malaria. So our project is to understand this evolution of the function of AKH from the corpora cardiaca to the seminal fluid. So our hypothesis was is seminal fluid AKH influence several processes in females, Aedes aegypti, through the regulation of gene. So this is our hypothesis. And to reach that hypothesis, first we decided to put in place two lines of mosquito, a mutant line and a wild type line, to better characterize the seminal fluid AKH. For that reason, we use a CRISPR-Cas9. We use a CRISPR-Cas9 to induce a mutation. You know very well CRISPR-Cas9, the two main components, the Cas9 and the guide RNA. So we use this system to induce a mutation in our wild type mosquito that we call LVP. So you see LVP in this presentation from Liverpool screen. It means also the wild type. The wild type mosquito. So we use the CRISPR-Cas9 to induce that mutation, and then it works well. So as you can see the difference between the wild type and the mutant. So the mutant has a deletion that is absent in the wild type mosquito. So in the wild type mosquito. So in our lab, you have two main lines. You have the wild type line and the mutant line. So we use the both to better characterize the seminal fluid AKH. So with these two lines, the first biological question was to know what is the impact. We know that what's the impact of the seminal fluid AKH on the feeding or the blood feeding behavior. This is just a quick remind you of the life cycle of the mosquito. So you have the eggs, the larvae, the pupae, and the adult. So this adult, only the females need to bite human because they need the protein from the blood for for the offspring. So that is the goal. So you can see, for example, this is an example of Aedes aegypti that try to bite someone. So in the lab, we give blood to the females to complete the life cycle, or to obtain the life cycle of mosquito. So you can see our system that we use. You have several systems that we use for the blood feeding. So we compare the female mated to the wet type. So it means that when I say female mated to wet type, it means wet type females mated to wet type, and then mated females mated to AKH null males. Also compare white type white type females and AKH null females. So you can see that we didn't see, unfortunately, we didn't see any effect of the seminal fluid AKH on block feeding and block digestion, but we did see an impact on white type females. We did see an impact of adipokinetic on the blood digestion. You can see, for example, you take this image, these figures. So on the y-axis, you have the curve of digestion, and then you have a different group that we use for comparison. So you can see that in general, the white type digest blood more slowly than the mutant on first day post blood mean. So after this result, we said, okay, we need to look the impact of AKH on remating. We know, for example, that one of the well-known seminal fluid molecule is a sex peptide, and this sex peptide prevents the remating in mosquito. Is it the case for AKH? What's the impact of the seminal fluid AKH on remating? And for that, we did remating. So we did first the first mating, white type females mated to the white type males, and then females mated to the AKH nouns. So during the second mating, we did the same thing. Female mated to the white type males, but this time the sperm was fluorescence. So we use the fluorescence males. Females mated to the AKH null males, and then the unmated was mated, because our idea was to check if there is a remating. So when I will call unmated, please remember that unmated means just one mating, and then we dissect to check the presence of sperm. So you can see on this result that when you receive just after just one mating, all the female were inseminated, but after the second mating, you check the wild type, you can see that you have none of them were inseminated. That's confirmed that yes, in general, females need some time after the first mating. The receptivity of the second mating is reduced, or the female become refractory to the second mating. But you can see that when AKH is absent, you increase the percentage of insemination. It means that the absence of seminal fluid AKH increase the probability of future insemination. So to confirm this result, we did the injection, just to make sure that to better understand what happened. We did the injection. Second time we use the male associated gland. The male associated gland is the tissues where seminal fluid are stored in mosquitoes. So we inject the seminal fluid and the normal AKH. So after that, remating, dissection. This time we just check, we don't use the fluorescence males because we use the injected male associated gland. So it was easier to make a difference between the first and the second mating. And then you can see this time when you inject the male associated gland, you have the same result. Wild type, you have a very low percentage of insemination. When AKH is present, you increase. And then we confirm that if you inject the AKH peptide, you reduce the percentage of insemination as compared to the control. The control is non-functional AKH. So it is clear that the female the female that receive AKH co-injection are less likely to remit than the control injected females. So because we see that, okay, the best way to see other phenotype will be by starting by the RNA-seq experiment. So we put this by starting with the RNA-seq experiment. So what's the impact of seminal fluid on gene expression? And from that, we can see other phenotype that are impacted by AKH. For that, we did this experiment. So we did female mated to the wild type, female mated to AKH norm, and virgin. The dissection, so the female was maintained on 3% sugar water. We dissect three tissues, the right of thorax, the abdomen, and the lower Purkinje tract at 26-hour post-mating and 24-hour post-mating. And then we did the RNA isolation and sequencing. You can see on the result that if you compare mated and unmated, mean that if you compare females mated to the wild type mosquito with the virgin mosquito, you have a lot of genes. You have several genes, and most of them are present in the lower Purkinje tract at 6 hours post-mating as compared to, you can see at 24 hours, you have a reduction of the number of differential express gene. But surprisingly, if you compare females mated to the wild type males with females mated to the AKH males, just one gene are significant differential express that we call the brain catinase and share. So this gene was upregulated in the female mated with the wild type males as compared to the female mated to the AKH nulls males. So that is so just to confirm our result, it was a great surprise for us to see just that one single gene. The question was maybe it was false positive, but that's when we did a quick PCR to confirm that upregulation the mated females in the mated females in the females mated to the wild type males. We know that in Aedes aegypti, the Wolbachia infection increased the expression of the brain catinase and share. You know that in the vertebrates, the catinase digests the cactin of insect in vertebrate, and then the evolution depends on food. So I hope that at the end of this presentation, I showed that there's an evolution of seminal fluid AKH, example of remitting. I showed that the seminal fluid AKH regulate the expression of the brain share catinase. Future study may consist of studying the impact of AKH evolution on insects and functional genomic analysis of the brain share catinase regulated by the seminal fluid AKH. So thank you for your attention. I thank you to our collaborator and the 4G agency, NIH, and the College of Worcester. Thank you.